Hello, everyone, and welcome to today's live broadcast, Quantitation and Profiling Metabolomics and Translation to Clinical Analysis, presented by Dr. Timothy Garrett, Associate Professor, University of Florida. I'm Alexis Krause of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by Labroots and sponsored by Agilent. Agilent is a leader in life sciences, diagnostics, and applied chemical markets. The company provides laboratories worldwide with instruments, services, consumables, applications, and expertise, enabling customers to gain the insights they seek. Agilent's expertise and trusted collaboration give them the highest confidence in our solutions. For more information on our sponsor, please visit www.agilent.com. Now, let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. We'll answer as many of your questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, click on the arrows at the top right-hand corner of the slide window. If you have trouble seeing or hearing the presentation, please click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Answer Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I'd like to now introduce our presenter, Dr. Timothy Garrett. Dr. Garrett received his undergraduate degree from the University of Georgia in chemistry, graduating Summa Cum Laude with highest honors in 1999. As an undergraduate, he worked in the lab of Dr. I. Jonathan Amster on the characterization of bacterial proteins using the MALDI-TOF, completing an undergraduate honors thesis entitled Improved Methods for On-Probe Cleanup of Unpurified Protein Samples for MALDI Time-of-Flight Mass Spectrometry. He received his PhD in 2006 from the University of Florida, working under the direction of Dr. Richard A. Yost. As a graduate student, he developed the first imaging mass spectrometry based on ion trap instrumentation through a partnership with Thermo, and studied the disposition of phospholipids in brain tissue. He is currently an associate professor in the Department of Pathology, where he is also the director of high-throughput metabolomics for the Southeast Center for Integrated Metabolomics, and conducts research in the application of metabolomics to disease diagnosis and clinical diagnostics. Professor Garrett is the author of over 60 publications and is an editorial board for clinical mass spectrometry. His current interests are in the application of direct tissue analysis approaches, as well as the use of high-resolution mass spectrometry in metabolomics and routine diagnostics. Dr. Garrett, you may now begin your presentation.
Great. Thank you very much for that wonderful introduction, and thank you for this opportunity today to talk about some of the work that we do in my lab and some of the work that we're hopefully trying to do in clinical diagnostics to sort of bring the next generation of diagnostics to our clinical lab. So today, I'm going to talk about several points. I'm going to talk about quantitative metabolomics. I'm going to talk about traditional quantitation aspects that are related to currently how we do most quantitation. I'm going to talk about where we can be going in the future in this approach. And then I'm going to talk about primarily the role of high-resolution mass spectrometry as it plays in quantitative aspects here. And then a key part of that is understanding isotope approaches that may enable different and multiple analyte analyses within a single run. So if we look at the traditional quantitative mass spectrometry that's done, it's primarily liquid chromatography, the LC, and MSMS, or tandem mass spectrometry. And so in this case, every sample gets an internal standard or a reference standard that helps you quantitate. Typically, it's a stable isotope version of that compound. For example, tryptophan D3 to quantify tryptophan. And in general, carbon is typically a better isotope than deuterium because we don't have any retention time shifts that might occur in a chromatographic run when deuterium is involved. But if deuterium is used, a D3 is usually preferred over multiple Ds in a sample. And so each metabolite is also externally calibrated, and we're covering the reference range of interest. And that reference range in a clinical setting is defined by the ranges in which it might be normal, abnormal, or out of the range of quantitation. And so one example would be, say, 10 nanograms per mil to 10,000 nanograms per mil. And each one of these has a defined LLOQ, or lower limit of quantitation, LOD, lower limit of detection, and a dynamic range that covers how linear the coverage would be in a sample. And each metabolite has a separate curve and separate internal standard and separate quantified sample for every metabolite. So that's if you have 50 analytes you want to quantify, you typically have 50 calibration curves to quantify each one. And the key here to point out is that we're measuring the ratio of the metabolite to the internal standard. And that's important because it helps to avoid ion suppression, and it helps to enable you to quantify in multiple kinds of samples where the ion suppression may be different across those different samples. So why would we want to do HPLC or LC followed by tandem mass spectrometry? Well, the primary reason is it helps us with the 3 S's in performance, and those are sensitivity. We have a really broad range from picomolar to micromolar range, and even lower than picomolar range. It's very selective. We have 3 ways to select an ion. We have the retention time from chromatography, we have the mass to charge value, and we have the fragment ions or product ions produced during that MSMS event. And the mass to charge value is one I'm going to focus in on a lot today because it involves the use of high resolution and high mass accuracy instrumentation to help push the boundaries of where we can measure within a single sample. And then the last S is speed. Mass spectrometry methods can be very fast. They can be less than 30 seconds, depending on how many analytes you want to measure. And so it enables high throughput analyses of thousands of samples in a week or a month, depending on the throughput of need. So if you sort of look at this in a figure, you can see that in chromatographic space, we can separate it along time, while in mass space, we measure the ions. And then the third window is MSMS, so a single ion produces a specific fragment that we may monitor to isolate that. So it allows us to be highly selective in a very complex matrix that would be in plasma or urine or serum, one that might be measured in a clinical setting. And this harkens back to a figure that was produced several years ago about the idea of combined techniques in mass spectrometry and how those combined techniques can enable improved measurement. And so this figure is an example of where you have signal that decays over time with a number of, not over time, with a number of stages of analysis. And you also have a chemical noise that reduces over time over the number of analyses. But combined together, those produce a higher signal to noise because then we reduce the noise at a faster rate than reduce the signal. And thus our signal to noise is what we measure in mass spectrometry. And so by doing 2 techniques, say LC followed by mass spectrometry, we get a gain in signal to noise, and then 3 techniques, we get a subsequent gain in that signal to noise. And so it allows us to have complementary selectivity. And so in most cases, you're doing LC-MS-MS, but you can do LC-MS-MS-MS or LC-ion mobility mass spectrometry, which would be a third complementary technique in separation. And one example of where quantitation is done is with a triple quad. And so what I'm showing here is an example of a schematic of a triple quad where we have 3 quadruples, quadruple 1, 2, and 3. In almost all cases, quadruple 2 is held at a constant value, just allowing gas to be introduced to cause fragmentation. So in this example, Q1 is held at a specific ion to say, let's say tryptophan is isolated. It's fragmented in Q2, and then in Q3, you can scan to find all those product ions to get all the different masses you might have interest. In a quantitative performance, you would select only 1 or maybe 2 of those analytes of those product ions in Q3 as a way to select a single reaction or selected reaction monitoring. When you're selecting more than 1, you might call it multiple reaction monitoring. And an example of this is a tryptophan, as I use across a lot of my figures here. tryptophan has in this case, on a low resolution, low mass accuracy instrument on a quadrupole, would have a measured mass of 205.1. We would isolate that ion and get a product ion spectrum on the second figure below, showing two primary product ions. One is a loss of amine to produce 188, and the other one is 146. And this points out a key part of selective reaction ion that is more selective than, say, a loss of a water or ammonia, because those losses are less indicative of a compound or a metabolite. So the ion at 146 would be a much more reliable SRM ion to monitor to ensure we measure tryptophan in a more precise way. And one thing to note here is in the figure on top, there's a lot of noise in the measurement. So it appears that we don't detect our ion very well, but in the bottom part, you see how clear the noise is. This is the reflection of multiple stages of analysis, reducing the noise to enhance the signal-to-noise measurement. And if we add chromatography in this case, we can separate, say, two other compounds or metabolites related to tryptophan. One is kynurenine, which is a downstream product of tryptophan through IDO metabolism, and is often measured as a way to monitor the immune system. And so we have a separation within that it elutes at an earlier time, the tryptophan. And the later one is a drug that has been studied to alter the immune system, called 1-methyltryptophan, or 1-MT. And so in this case, we can monitor this drug, 1-MT, and then two metabolites that might be altered by that drug performance to get a general idea of how the body is reacting in that given instance. So if we look at a different example, one that is a smaller or maybe more polar analyte, metformin is a very polar polyamine drug. It's used in the treatment of type 2 diabetes. There's also been some recent reports evaluating it as a longevity drug, although that's still under works. And so in our case, we wanted to look at the evaluation of metformin in relation to a lupus or a autoimmune disorder that was studied to see whether or not we could quantify values in stored samples to then develop a future project to evaluate the use of metformin in larger clinical trials. And so in this case, we developed a standard LCMS method. And in our setting, we use a HILIC, which is a hydrophilic interaction liquid chromatography, as a improved way to do separation because it enables the retention of more polar analytes rather than reverse phase chromatography. And so a quick example of this is, say we took 20 microliters of the patient sample and added acetonitrile containing formic acid that also contained the internal standard. In this case, we're using a D6 metformin, the one that was only available for this setting. And then we mixed it, we centrifuged it, and then transferred only the supernatant. So you can see it's a very dilute process, adding 500 microliters to only 20 microliters of patient sample, and then only injecting 2 to 5 microliters for analysis. This is around 350 microliters a minute. And if we look at a simple example of 10 nanograms per mil of LCMS method on a triple quad, you can see the separation where the retention is fairly good for this analyte for HILIC, and with a mobile phase described as above. But the point also, because this was an LCMS QQQ run, can we possibly use this as a high resolution approach to monitor metformin? Because part of the goal would be to also monitor other analytes present in a sample rather than just the drug of interest, but other metabolites that may be affected or may help us understand the phenotype of an individual taking this drug. And so you can sort of see here that that's a calibration curve that looks fairly linear in this case. And so now switching to high resolution, we also decided flow. So now we're throwing two things at it: a low flow separation HILIC approach using a different source. And the same separation characteristics, with about 30,000 mass resolving power and collecting a wider mass range of, say, 70 to 1,000 in positive ion only. And this would allow us to get the endogenous metabolites, but also target the metformin compound of interest. And this is that translated example running at 1 microliter per minute And this is a 10 nanogram per mil metformin separated with about a minute only on this. You get in HILIC at low flow rate, you get a little bit less retention than you would at higher flow rates in a larger bore column, but it still enables us to measure this compound of interest. And you can see the product ion spectrum below, measured down to the mass accuracy of less than 5 parts per million, and getting product ion spectra that allows us to validate that this is the right compound of interest. If you look very close down, sorry, that, excuse me, I'll go back to that slide before. That wasn't the product ion. The point I'm trying to point out there, this is the background in a high resolution scan. And the next slide, this is where metformin is. So it's really down around 10% of the signal, and it's in what you consider the noise, but of a typical mass spectrum. But now we have very clearly isolated the compound using high resolution and high mass accuracy. And then if we look at a calibration plot, we have a triple quad on the right, calibration, and then high resolution quantitation on the left, looking at a very similar performance, maybe at a little bit higher error on the higher end of measurement. But the key is really knowing how well the methods perform against each other. And so if we look at the quantified performance between the two methods, and there's only high resolution, no tandem mass spectrometry. So just using the mass accuracy of the instrument and the resolution of the instrument to isolate the ion. And so in this case, we get almost a less than 5% error in most cases. And when we don't measure it in both methods, which is reliable. And then maybe as high as 6% in this quick patient sample. And then the QC of 500 looks very reproducible across both methods. And so what this tells us is that we can run a very similar quantitative performance on both types of instruments for a target compound with a wide range of interest. And this allows us to then do possible future studies that enable us to do endogenous quantitation of analytes of interest beyond just the one targeted for this drug compound. So that leads us to what we want to do in the future. And so in traditional untargeted metabolomics, which is what I do a majority of my work in, we are typically doing an experiment without really knowing what the significant features are, because that's the point. We're trying to develop new significant compounds or new significant metabolites related to disease, versus in targeted quantitation, where we would look for targets of interest and then try to see if those are significant between the samples. And in a combined workflow, we would run maybe untargeted first and then targeted quantitation second on a set of compounds of interest. So we're then doing two different experiments on the same set of samples, which would use more samples or use more material and use more instrument time. And then in some cases, and I'll show you an example later, that sometimes targeted metabolomics is chosen first because results are easier to interpret. And that's just part of the field of metabolomics as it grows and as we make metabolomics easier to understand. But our sort of goal is really to be able to do combined quantitation in a single injection with high resolution mass spectrometry and getting an internal standard for each compound. And in the next few slides, I hope to show you what that really means. And so really as sort of a background, is kind of just show you what this conceptually means. The only thing that we're really changing in is mass spectrometer, and that's really the improvement of how we measure the compounds of interest. So really just measuring the mass in a more precise and a more highly resolved way. So you can keep the same chromatography, the same column, and the same flow rate without any things that are changing. The only other and the additive benefits are here described below with that mass accuracy and combining that with resolution. And of course, you can add MSMS, and so you're getting mass accuracy and high resolution in MSMS mode as well. If you look at just a specific example, and most of our work is done on a time of flight range, but this just shows you the types of instruments that would be available for this kind of work. A quadruple, which is what we started our work on, doing MSMS or isolating a compound of interest. You have fairly low resolution. You can tell differences in this figure. And then when you increase the resolution, you're going to see more of the picture, right? And FTMS is the highest resolution available on the market, but you typically would need much more expensive instrumentation to do that work. and enable that clear separation. But with a time of flight or with a other kind of measured instrument, you get an improved measure of that compound. And it just, well, ideally, it's really just improved clarity of the measurement. And so this is really an example of looking at one compound, valine, a common amino acid that might be detected in the sample. But you can look at the carbon 12 ion of interest versus the carbon 13 ion of interest. And the key part here is you can measure the abundance of both. And so measuring the abundance of the carbon 13 allows us to know how many carbons are present in that compound. And so in this case, our measured abundance of the carbon 13 isotope of valine is about 5.4%. And the actual abundance is 5.5%. So we could get a very good estimation of the number of carbons within this compound if we have enough resolution to separate it out, the isotopes out from the carbon 12 isotope. But if we look even closer, there's more information there depending on the resolution that might be available or how high you can measure. You can even separate carbon 13 from nitrogen 15 isotopes to help and better inform your prediction. So this is just one of the added benefits of high resolution mass spectrometry that might enable you to, as an improved measured mass and improve measuring your compounds of interest. So now let's take a different example where I've talked a lot about tryptophan. I've been studying tryptophan for a while in terms of the metabolic pathways of tryptophan for initially with organ transplantation and initially with just looking at tryptophan to kynurenine as a measure of whether or not we can use that ratio to help predict organ transplantation, whether it would continue in a successful route or whether we'd have to bring the patient back in to understand whether or not they're rejecting that organ. And in general, that is a fairly good measure in that setting to be able to monitor how well the immune system is working and whether or not you're in an activated immune system or whether or not you're working in a normal immune system environment. But ideally, tryptophan has other metabolites of keen interest. One, serotonin is another one of interest, as well as melatonin. Both of those are neurotransmitters that do have different neurological states. Typically, melatonin is associated with sleep, and serotonin is associated with an overall feeling of joy or feeling in a fairly good mood. And you have other downstream products that are part of the kynurenic pathway that might have other neurological effects that would be important to measure. And so it would be very important for us to be able to measure as many tryptophan metabolites as possible so we can not only measure immune system, but also measure the neurotransmission parts that tryptophan is related to. And one thing that I haven't pointed out yet, but tryptophan is an essential amino acid, so it's something that we do need to get in our diet to be able to provide the tryptophan-related pathways and ensure that we're getting the right metabolites in our body. And this is an example of, say, looking at tryptophan, looking at the separation of tryptophan at one specific mass resolving power. And this is tryptophan, target compound at 205.097. And then in this case, we had more than one internal standard. We had one at 208, which is a D3 deuterium labeled. And then we have a carbon 13 labeled at 216. And so with this, we get accuracy, we get the isotopic envelope, but we can also get some other features if we do, say, isolate both of those ions or all of those ions of interest. And this is an example of just sort of how high resolution can be used as a further separation mode. So in a typical triple quad kind of way, you might isolate each one of the target compounds of interest. So I'd isolate tryptophan separately, then I'd isolate its internal standards separately, and I'd measure those transitions, which is a great way to do the experiment. But with high resolution, we could possibly isolate a wider range of those masses and include all the masses of interest in a single scan. And so in this case, I show you what that might look like for tryptophan, coloring the ones related to each other together. And so we would have and you can sort of see how well those separate out and how well the MSMS still performs, where we might isolate, say, a 10 AMU wide window. And this is collecting all of those product ions and using the product ions from that as a way to quantify. You might see how well this works, say, for MSMS. In this case, this is MSMS of each one of those compounds. So it shows very nice performance along with its internal standard. And in this case, we had an internal standard for every compound And that very good signal to noise for a good quantitation measurement. But now you can look at just the mass accuracy, and that's just using the full scan MS, no MSMS, with a 5 ppm mass accuracy window. So this is still targeted, but it's targeted using the mass accuracy of your instrument as the selection window. And you can see we still get very good signal to noise in most cases, maybe for the standard for anthranilic acid, which would be anthranilic acid IS as the internal standard. But in most cases, we get very clean chromatographic performance. So this is a method that we now apply. We use this quantified method for many different studies. And one study I'll show you as a highlight is tryptophan metabolism. So using an understanding of what our gut and the role of our gut is in doing tryptophan and metabolizing tryptophan within the body. And so this is a collaboration with University of Florida professor Dr. Gary Wang in infectious disease and global medicine. Our main goal here is to look at germ-free mice. So those are mice without any microbes in their gut. Look at a set of mice that have a single type of microbe in their gut, and that would be a monoassociated E. coli. And then those that have a tryptophanase depleted, where no tryptophan is present, or no species are present to degrade tryptophan, so we can get an understanding of that. And for now, I'm really just going to show you the germ-free results. So because we can want to, as we're still building this project. And our main goal, and one of our main goals, is really to focus on understanding the role of a specific nutrient, in this case tryptophan, on the role it plays in gut and the gut-brain axis, and how that may relate to how we would improve our neurotransmission or improve different parts of the tryptophan metabolism within our system. And if we look at a single example, this is looking at germ-free mice and looking at, or germ-free rats, excuse me, with a clear separation on the left. If you're not familiar with metabolomics of statistical analysis, this is principal components analysis, or PCA. And the PCA plot on your left shows the separation of control and tryptophan supplemented rats from those that are deficient, where we get a very clean separation between the deficiency and those that had either the normal amount of tryptophan or of a higher amount of tryptophan. And then the depleted ones are in green. And really basically what that shows you is that when you deplete an essential nutrient, you're going to have a very drastic effect on the system, because that your body does not have that system, or does not have that nutrient to make the key metabolites of interest. If we look on the right, this is a heat map, or a clustering map, showing the top 25 metabolites that are present in this sample. And in this case, this is serum isolated through protein precipitation, where we would remove all the protein, isolate only the metabolites, and then inject the small amount. This would be injecting about 5 microliters of a sample onto the chromatography system. And in this setting, we're using reverse phase chromatography as the separation. And so when we look at a heat map, we can get a better idea that we do separate out some metabolites that are key in the tryptophan supplemented and those that are normal. And some of those would be, of course, serotonin, which is at the bottom. Xanthranic acid at the bottom, and tryptophan. Obviously, when you're increasing tryptophan, you have some differences. But another key metabolite in this case is serotonin, that's highly increased in those that are supplemented with tryptophan. If we look at the pathways that might be highlighted in this case, you can sort of see that where we have tryptophan as a key nutrient leading to serotonin and acetylserotonin, and down that neurotransmitter pathway, versus also down to the nicotinamide or niacinamide pathway, leading to NAD and NADPH. So in a summary of the tryptophan approach, we use this method all the time. We quantify methods with high resolution only and get all the other metabolites of interest. And if you saw in that heat map, there's more metabolites, obviously, than we quantify. We only quantified about 6 in that measurement. And so ideally, we'd like to measure more than that, maybe 500 to 1,000 different metabolites, so we can get a better quantitative performance to know how much is there versus how much is missing, or how much is indifferent. In that case, how much is deficient. So one of those approaches that we're looking at is using isotope labeling, approaches that grow cells and grow cells with a high amount of carbon-13. In this case, we're using 95% carbon-13 in a technique called isotope ratio outlier analysis, or AROA. And this is in a collaboration with AROA Tech. And if we look at what's here, in the typical, or on the left side, you have a carbon-12 side. This would be a glucose, carbon-13 glucose fed at 5% labeled, and that would produce a descending isotope pattern that increases the carbon-13 abundance by a significant amount, so we can measure it better. When we combine both of these together with the 95% carbon-13 and the 5% carbon-13 labeled, we get this pattern that produces a U-shaped pattern that produces a non-natural isotope effect. And so on the right side is the non-natural aspects, where we have an M-1 and -2 that would not be normally present in the given system. So what I sort of tell people is it represents the DNA of a metabolome, because now we're producing things that are not natively present, and we can easily capture and identify metabolites that are isotopically labeled through glucose in a given system, and then match those with the carbon-12 side as a way to measure the single metabolite with both an internal standard on the right and the natural abundance on the left. We accomplish this by using yeast as a primary test case, because it's a little bit less expensive to use and it's fairly easy to grow, and a lot of conditions enable us to produce it in high quantities, which is what we would need. And so in this case, we extract only the small molecules from the yeast, and we standardize everything to that extract. And we're using the yeast extract essentially as the internal standard. It's 95% labeled, and it would allow us to do both normalization and quantitation in the same run. And this is an example of what it would look like. So in this case, we have the natural abundance of tryptophan, where they have normal carbon-13 abundance on the left with carbon-12 and carbon-13. And then on the right side, we have fully labeled tryptophan with that negative isotope pattern, producing that very distinguishable feature that allows us to identify it in a complex matrix. And in this case, we also can then calculate the exact number of carbons. So for an unknown metabolite that we might not have identified through the various means, we can use the carbon-12 side and the fully carbon labeled 13 side as a third measure, because we still get the isotope of the single isotope carbon-13 label as one measure, but as a third way to confirm the number of carbons that are in a specific unknown, which allows us to then do better formula prediction. And so this is an example of what it might look like when you're using a low versus a high concentration of a target compound. In this case, it's histidine. So looking at a higher amount where we spiked it in above our internal standard. In this case, it's the highest level. It's at 62 micrograms per mil versus at the lower level. And you can see that performance is the same normal performance you would get with a quantitative run, keeping your internal standard constant. And we still have the same preserved isotope pattern for everything. And if we look at the linearity that might produce in something like this, it follows the same lines that you saw earlier in the slide, where triple quad quantitation with an internal standard, the gold standard of what we would measure everything against, versus what I showed with tryptophan, high resolution MS or high resolution MS quantitation, very linear calibration curves, and very, very strong sensitivity. You can even do both reverse phase and Hillock approaches to separate and quantify the same compound. And in this case, I'm showing proline, which is retained longer on a Hillock approach with a little bit higher RSD than we might get on a reverse phase approach. But those things can be measured and optimized depending on your system. And in biological samples, you can do the same thing. That was just looking at a bunch of standards. And here you can see this just shows what it would look like with tryptophan again. And again, that change in response that you might see. And then again, showing another amino acid, not just ornithine, but other amino acids are of course quantified well within the system. And this is looking at threonine across multiple runs and how we might look at the reproducibility on a given day or multiple or a given week. And so in the end, we're still working on this method. We've been able to quantify most of the amino acids using this approach and expanding out to other compounds of interest as a secondary validation. And so recovery is very good. This is within plasma samples. Of course, we run a very rigid validation and quality control with each run, very low variance. Stability of the yeast is so far very stable, so it allows us to store it for quite a bit of time for measurement. And again, we're measuring the natural and endogenous natural abundance against an internal standard. And in general, we have what we should have in yeast extract is somewhere around 500 different metabolites. So we have the ability to expand it well beyond what we might measure. And this was only doing high resolution, no MSMS, no further quantitation, but we're still validating where you might need MSMS and where you might need only high resolution as a key future component of doing quantitative analyses with this kind of approach. And so ideally, what we'd like to do is be able to provide a quantitative performance on this platform using all of those key target compounds that are in there. But we haven't lost our ability to look at unknown metabolites that might show up in unique samples. So we can still use data mining and tools that you might use for that, such as open source tools like MZMine or other tools that you might use to do statistical analyses. You can look at many different ways to identify those unknowns to help fill in the metabolomic space. And you can also look at what future trends might be by capturing a lot of different unknowns in a single run. And sometimes it's easier to just look at the targeted analyses and gather, say, quantify a small set of targeted compounds and only look at those that are known, because it is, in general, it is an easier way to provide results to individuals. So why do we want to still do untargeted? And here is sort of an example of what I like to show. And that is that we have a global metabolomic run on the left. This is looking at prostate cancer and looking at PCA of, say, 2,000 different compounds versus on the right, where you're looking at PCA of a set of around 50 different analytes. So individually, the analytes there are significant metabolites within the targeted quantitative set, but overall, when you look at them as a group, they don't perform in terms of being able to separate the manifestations of a clinical disorder. In this case, this clinical disorder would be not just prostate cancer, which would be here in the green, but also prostatitis or benign prostatic hyperplasia, BPH. And ideally, measuring clinical samples should measure the clinical scope or clinical manifestations of a disorder, not just between cancer, but between other disorders that might be present. So you have a test that might improve the quantitative performance. And so if we use a profiling approach, we can capture more metabolites and get a better idea of those that might separate our disorders of interest rather than just looking at a smaller quantitative set. And this just shows you that in the same example. So some things to summarize. There are still no sort of rules that exist for how accurate you need to be. And so those kinds of questions need to be validated. So do you need to have 5 ppm every time, or is 30 ppm mass accuracy sufficient? In some cases, the only way to test that would be to look at how reducing your accuracy and how well your method still performs. Again, that same goes for mass resolution, and that's, of course, instrument dependent and metabolite dependent, but you need to know when your method may not work depending on when the resolution falls apart or when isobaric compounds are interfering it and don't allow you to do better mass separation. And then the dynamic range is a key part. Typically, you have shorter dynamic range with high resolution mass spectrometry than you do with a triple quad. And so you have to take that into account when developing a method, that your measurement range needs to be accounted for in the instrument in which you use to do that quantitation. And so with that, thank you for your time and listening to this presentation. And I also would take any questions that you might have.
Thank you, Dr. Garrett, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Answer a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. So let's get started. Our first question is, why did you employ high-resolution mass spectrometry for quantitation and metabolomics, since it is usually performed by a triple quadrupole?
Well, in our case, using high resolution was a key part to help understand the future of what we want to measure. And so one of our goals is to be able to use a single platform to do many different kinds of measurement, rather than having to expand a new instrument every time we need to do a different measurement. So in this case, using high resolution enabled us to separate out, at least in the ones I showed, separate out metabolites of interest using only high resolution and mass accuracy, and get similar performance that you might get with a triple quad, as well as being able to improve the identification of other species within the sample, within the set that you may not have thought about measuring or may not know about measuring. And so really, in this case, it's one that we really want to expand the role that high resolution might play in a clinical setting, knowing that most clinical labs and most clinical research for quantitation is done on a triple quad. So it is an uphill battle to try to do that. But I think with the right examples, I think hopefully we can be shown that it has the capability to do it right and precisely.
Our next question is, why did you only show amino acids for your quantitation with labeled yeast extract?
So far, the amino acids is just what we focused on as a way to do, say, interlab studies. And it's a good start to be able to do it, because if you can't quantify the amino acids within a run and within a metabolomic run, then you probably won't be able to do other analytes of interest. So they represent a good set and a very well-characterized compounds that allows us to develop both reference ranges that are consistent and measurable against clinical samples, and then also apply them to different studies. And so we don't want to just sit with amino acids. We will expand it to other ones, but as we do the validation, it's a good starting point for that aspect.
And Dr. Garrett, it looks like we have time for one more question. In your tryptophan study, you showed a KEGG pathway with some key metabolites highlighted. Does your approach detect all the tryptophan metabolites?
That's a good question. And no is the answer, because some of them are below the limits of detection. And as well as some of them are sitting in a noisier area depending on their mass resolution. And so we're looking at ways in which we might be able to decipher more of the tryptophan pathway and use this study as a way to do that, because it represents a very unique way to validate a metabolic response or a very common way to validate a metabolic response, because you want to increase a key nutrient. And by doing that, you should be able to highlight the entire pathway and know whether or not you're measuring everything within that pathway. And so one of our goals with this study is to go back and look at limits of detection for other key compounds and how we might have to concentrate the sample or do different sample preparation in order to highlight different components of the metabolic pathway for tryptophan. And hopefully in the next few months, I'll be able to show you how that would work.
Thank you again, Dr. Garrett. And do you have any final comments for our audience?
The only final comment I would have is I hope that I've showed you that using high resolution quantitation is possible, and that you may, in your next experiment, take the opportunity to try it and see how well your method could work in that setting.
Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Just a reminder, any questions we did not have time for today will be addressed by the speaker via the contact information that you provided at the time of the registration. We would like to thank Labroots and our sponsor, Agilent, for underwriting today's educational webcast. This webcast can be viewed on demand through November of 2018. Labroots will alert you via email when it's available for replay. Encourage you to email with your colleagues who may have missed today's live event. Until next time, goodbye.